Sue Romanoff from Edison Group. Today, I have the pleasure of catching up with Eric Manting, CEO of Mendus. Hello, Eric.
Hey, Sue, how are you?
Great. So your lead asset, Vididencel, looks like a promising candidate for AML and ovarian cancer. Could you provide an overview of how the vaccine works and then maybe potential advantages over off-the-shelf therapies?
Absolutely. Yeah, I think it's actually catching two questions in one, Sue. And one is, how does this product work? Let me start with that, and then also explain what it means in terms of manufacturing. There has been a lot of experimenting in the past decades in combining cancer cell biology with dendritic cell biology. And the simple reason is that cancer cells are very good in evading the immune system, and dendritic cells are basically orchestrators of the immune system, so they are very good in stimulating the immune system. Now, to combine the two results in a product that basically feeds the immune system with cancer cell information, which is what you want if you want to trigger an immune response leading to, in the end, an immunotherapy against cancer. That is how also Vididencel works. So it's actually a cancer cell that expresses dendritic phenotype, and that makes it highly immunogenic. And it's a black and white phenomenon. So the cancer cells, also our starting material, are ignored by the immune system. But as soon as during manufacturing we shift these cells to express dendritic phenotype, they become highly immunogenic and they start stimulating the immune system. So that's how the product works. And what we have discovered, and also, of course, documented in detail, is that it actually stimulates the antigen-presenting cells of the host. So in other words, we inject Vididencel product in the skin, and the skin is an environment which is very rich in antigen-presenting cells because it is a natural barrier to entry, for example, against infections. So as soon as we inject the product in the skin, local antigen-presenting cells see something is wrong. They start phagocytosing, so eating our product, the Vididencel, and then basically train the rest of the immune system to build up an immune response. They do that in a specific way. They crawl out of the skin, basically are moving towards the lymph nodes, and that's where they locally train T cells and other immune cells to act against this strange invader which they have just picked up. So that's how the product works from a, let's say, basic biological principle. What's very important for us as a company is that we can manufacture the product and that we can do it in a scalable and in the end also economic way. And what has been holding back the field largely, everything related to dendritic cells, is complexity of manufacturing. And that has to do, on the one hand, with the fact that these cells are very sensitive. They're not called dendritic cells for nothing. So they have dendrites, they have, let's say, a sticky nature, which makes them difficult to work with. But also, if you start with dendritic cells and you still have to bring in the cancer cell antigens, then you have to find those antigens also somewhere, right? So if you start with, for example, patient dendritic cells, you have to harvest them first, and you have to load them with the cancer antigens. And all of that is a rather complex way of making the final product. And of course, then it also still has to find the patient as a treatment. Now, what we do is we have one product in all. It means that we grow large quantities of leukemic cells. They are our cells. It's a proprietary cell line with GMP grade cell banks. So it's good manufacturing practice grade cell banks. And these cells allow us to make a lot of product, which in the final stages of the manufacturing process, we shift from the leukemic phenotype to the dendritic phenotype. And then you end up with this highly immunogenic product. We irradiate the cells for safety purposes. We store the cells frozen. So for patients and for hospitals, it's actually available on demand. So as soon as a product is needed, it's available in the hospital. It can be thawed by simple bedside thawing, and it's administered via intradermal injection. So for the patient, in the end, it's a very straightforward product. And also for us as a company, this makes this a scalable and in the end also very efficient way of making the product large scale.
So your approach to target AML is really in the maintenance setting, which is really an unmet need, right? I mean, could you recap highlights to date and the key takeaways that we should expect in December for your Advance 2 update at ASH?
Sure. Well, I think what makes a good maintenance treatment is actually two things, which is one, a product has to be safe because maintenance is basically a therapy that follows initial chemotherapy. And AML, that's very harsh chemotherapy
Yeah.
because the disease is so aggressive that the first thing you have to do is to bring it down as quickly as possible and get patients in a complete remission. But then you cannot treat with toxic products on a continued basis. You have to find a safe product that allows you to continue to treat patients, but that does not burden the quality of life and the health of the patient. That is what our product brings, and that's why it's also suitable for maintenance therapy. It's a very safe product. The only side effect we've seen is actually where we inject the product, where the skin shows signs of inflammation because the product is doing what it's supposed to be doing, which is recruiting the local immune cells. But after a couple of days, that redness disappears, and it's the only side effect we've seen associated with the product. The other very important part in maintenance therapy is durability of response. And it means that not only you see that patients act short term to the product, but that they're building up an immune response that allows them to keep control of the disease over a longer period of time. Now, if we go back to the start, we had a phase 1 trial that clearly showed a dichotomy. Either patients already had high circulating levels of cancer cells, and they were on their way to what's called a florid relapse. So it's a very quick burst of AML. There's no way stopping that with a vaccination-based approach, actually any immunotherapy that we know to date. You simply can only kill those cells with high doses chemo. If the patients were, however, with low numbers of blasts, we saw a very good response. So from the phase 1, we already learned that we have to focus on those patients that have low levels of disease. That was the basis of the phase 2 trial, which is currently ongoing and on which we will provide an update at ASH. So those patients are in what is called a first clinical remission. That's after they've undergone high-dose chemotherapy. But all of these patients that were recruited in our trial were still positive for the disease in what's called a so-called MRD, or measurable residual disease, a positive state. MRD is associated with very fast relapse. Sometimes it's a matter of months. And this is the patient population that really requires maintenance therapy, right? And that's the patient population we treated in the Advanced 2 trial. End of last year, we showed for the first time that the majority of patients was still alive at the end of the trial, 14 out of 20 patients, and also that 12 patients remained alive without further treatment, ranging from 16 to 48 months after start of treatment with Vididencel. This year, we will, of course, provide an update, a survival update on where the trial currently stands. And what we have also done and been able to do in this year is to collect the so-called immunomonitoring data. And the reason is a technical one. To do these immunomonitoring assays, you first have to collect all the samples because you have to open them all at once and you have to do all the assays at once to get a good consistent picture on a per patient basis. So only this year we had collected all the samples and we've analyzed them in a lot of detail. So also this year at ASH, we will present 2 abstracts on the immunomonitoring data, showing what the product does in the way we just discussed, right? So triggering strong immune responses first in the skin, and then you can pick them up in the blood. And what we also are able to show now that we have all those data at hand is the correlation between the two. So how the immunomonitoring data and also patients with a very strong immune response also have the best clinical benefit from the product. So I think all in all, a rather big data package has come together, and we will present an update this year at ASH on all those different data we collected from the trial.
Great. So for oncology combinations are really promising. So I think it would be helpful to understand your plans for your upcoming trial. And then also, what should we expect? And then how do you anticipate it will influence the clinical development for your therapy coming up?
Sure. Yeah, I think it's a tactical point at a certain moment when you have monotherapy data, which is quite unique in the cancer treatment landscape, which is very crowded. Where do you go next, right? And in AML maintenance, there are certain settings where there's currently still no drugs, so where you could also do a monotherapy trial. But we thought it was better to focus on the broader AML maintenance setting and basically follow the label of the only approved drug currently in AML maintenance, which is called Onureg, which is the brand name for oral azacitidine. So the first thing we will do is to combine in a larger trial with oral azacitidine. And because it's then no longer monotherapy, we of course will also build in a control arm. Right? And what we will do, and details will be disclosed shortly, we are of course in full preparation of the trial. So as soon as we are done with all of that and we can announce it, you will see the details. But what we will aim for is a two-stage trial, whereby we first focus on a smaller number of patients, again with a control arm, but to show safety and hopefully also some initial signs of efficacy. And then if that first part is completed, we can expand the trial to a much larger number of patients. So that's, let's say, the trial that we are currently preparing for, again, in combination with standard of care being oral azacitidine. What it means in the bigger picture of developing this product to, in the end, market registration is twofold. First of all, we continue to build out the data package around the product. Secondly, we are building up a global network of clinical centers. What you will see is that we will do this trial outside of Europe. The ongoing trial is with 10 clinical centers throughout Europe. This trial will be outside of Europe. We also have the IND open in the US. So as part of, let's say, the global registration strategy for the product, we will also look to build out a global network with clinical centers. And that's an important step that we will take with this next phase 2 trial. Independent of the launch of the phase 2 trial, we will also start preparing for a global registration trial, right? And that is something that will be supported by the new trial, but which is not absolutely dependent on the new trial. I think also from the already ongoing trial, which has completed its active phase and is now a long-term follow-up, so the monotherapy trial, we will have very substantial data package to discuss with the regulatory authorities. So in parallel to setting up the new phase 2 trial, we will of course also start planning for the global registration trial.
So it looks like you're gearing up for a new trial for your second asset. Could you provide context to the data we've seen so far, and then what a clinically meaningful result may look like?
You mean for ilixadencel, the
Yep.
other product? Yes.
Yep.
Yeah, good question. So with ilixadencel, we've learned a lot of lessons. We've done quite some clinical trials with the product. For those of you that are not familiar with the product, it comes from a different source. It comes from healthy donors. It is an allogeneic product, so we can make multiple batches of product from one donor. We are not dependent on patient material, and we administer this product intratumorally. So we inject it into the tumor microenvironment, and that will switch the microenvironment to a more immunogenic one, and hopefully that will, of course, lead also to long-term clinical benefit. What we have completed is a larger trial, a phase 2b trial in renal cell carcinoma. And what has happened there, and that's a lesson learned, I think, not only for us as a company, but also for the broader immuno-oncology landscape, is that checkpoint inhibitors, which have become a very successful class of new cancer therapies, have basically taken over quite a lot of tumor indications as first-line treatment. So also in this case, in the case of renal cell carcinoma, checkpoint inhibitors have become standard of care. And that was not working out well for our product because it did show clinical effects, but not to the extent that it was superior to the checkpoint inhibitors. And then as a company, you have to take a tough decision, which is, will we continue to invest in this direction or shall we look for a new direction? We've chosen to do the latter, and also to look for indications that are not responding to checkpoint inhibitors, to not, let's say, fall into the same trap again. And we have looked into all the data we collected, not only in renal cell carcinoma, but also in a broader range of solid tumors. And we saw interesting data in a selected group of tumors called gastrointestinal stromal tumors, which is a subgroup of soft tissue sarcomas. So what we have decided to do is to move that program forward in the broader class of soft tissue sarcomas. And we are currently in preparation of a new phase 2 trial, which will allow us to move that product back into the clinic. And of course, we'll disclose details as soon as we can. So
as you wrap up a really busy year and then kind of look to the next, What are the key catalysts investors should look for in the next 12 to 18 months?
Good question. I think first and foremost, it will be ASH 2023. Again, because we collected so much data out of the ongoing phase 2 trial that it will be a very important update for us to not only show the survival data, but also how the survival data relate to all the immunomonitoring data we have collected in the course of this year. Then, of course, we have the launch of the new trial and the details of the new trial to look out for. We will have the launch of the ilixadencel trial that we just discussed. And let's not forget, we have an ongoing phase 1 trial in ovarian cancer. And ovarian cancer is an interesting one because it's very tough to treat. Checkpoint inhibitors so far have not been successful. And it is a tumor that requires maintenance therapy because after first line of treatment, the majority of women have the disease back in a very short period of time, mostly within the first year and certainly within the second year. So also that indication is one that is really interesting from a maintenance therapy perspective. And we have just announced at SITC, the Society of Immunotherapy for Cancer conference, positive initial data. So we have shown a very strong boosting of immune responses in ovarian cancer patients. And we have just disclosed that we have enrolled 16 out of the 17 patients. So that trial is also ready for complete enrollment this year. So also in the course of next year, that trial will continue to produce additional data. So short term, I would say it's ASH. Midterm, it will be the launch of the new trial in AML, the launch of the new trial in ilixadencel, both trials, of course, also producing data in the next 12 to 18 months. And let's not forget the ovarian cancer trial. So I think we have plenty of milestones ahead of us too.
Right. Sounds like busy calendar. So thank you, Eric, for helping us better understand your company and all the work that you're doing. Thank you all for joining us here today. If you'd like to learn more about Mendus, please refer to edisongroup.com. Thank you.